Today, we're diving into how scientists are learning to read the most complicated, most important book ever written, the Book of Life itself. So just imagine for a second. Someone hands you this enormous book, and it's got 3 billion letters inside. It holds all the secrets to life, but it's all just gibberish to you. You can't make out a single word. Well, that's exactly the problem scientists face when they get raw genetic data.
See, we're in the middle of this massive data explosion. We've gotten unbelievably good at sequencing, at figuring out the order of all those genetic letters. But knowing the letters isn't the same as being able to read the words.
I mean, thanks to some incredible tech, sequencing a genome is now super fast and pretty affordable. We can get the raw sequence, that endless string of A's, T's, C's, and G's, for just about any living thing. So now we've got all these books of life just sitting on our servers, which, is a fantastic problem to have. But it does raise the question, how on earth do we make sense of it all?
And this is where the magic really starts to happen. To turn all that raw, meaningless data into something we can actually use, we need a translator. And in genetics, that whole process is called annotation. So, what is genome annotation? Well, it's exactly what it sounds like. It's the process of taking that long, mysterious string of letters and literally adding notes, annotations to it. It's how we find the genes, how we figure out what they do, and how we identify all the other important bits and pieces hidden in that code.
And this slide just shows the difference perfectly. On the left, you've got the raw sequence. It just looks like a random string of letters, right? But after annotation, look at the right. It transforms into something meaningful. We now know that specific bit of code is gene A, and its job is to produce a specific enzyme. It goes from a totally meaningless code to a readable instruction manual.
Okay, so how do scientists actually do this? How do they perform this translation? Well, they've got a whole set of really powerful tools and a pretty clear process they follow.
The whole process is kind of like detective work. First, in step 1, you've got to clean up the data by finding and masking all these repetitive DNA sequences that can kind of confuse the analysis. Then, step 2, you use computers to predict where the actual genes, the important clues, are located. And then, the final, most important part, you figure out what those genes actually do. You assign them a function.
And one of the key tricks here is something called homology detection. It's actually a pretty clever shortcut. Think about it. If you find a sentence in a language you don't know, you could try to translate it by comparing it to a huge library of books that have already been translated. Scientists do the exact same thing with genes. They take a new, unknown sequence and compare it against these massive databases of known genes. If they find a really close match, they can pretty safely infer what its function is.
So why do we go through all this trouble? I mean, what's the payoff? Well, it's because annotation is absolutely fundamental to modern biology. It's how we unlock some incredible genetic secrets.
And this is the main takeaway, really. Without good annotation, a genome is just a big, useless file of letters. Fields like proteogenomics, which looks at all the proteins an organism makes, completely depend on it. I mean, you can't possibly understand the final products if you can't even read the blueprints, right? And the kinds of treasures we find are just amazing. Annotation helps us find things like biosynthetic gene clusters, which are these little molecular factories that might create the next generation of antibiotics. Or we find special enzymes, inhibitors that control key processes, transporters that act like gatekeepers for cells, and even toxins. Every single one of these is a story just waiting to be told.
But for all the amazing power of these computers and fancy algorithms, the job isn't quite done. There's one last, absolutely essential component.
And this quote right here just nails it. The automated tools, they do a ton of the heavy lifting, but they're not perfect. For the really important genes, the ones that might be totally critical for a particular species, you need a human expert to take a look.
And that's really the final step in this whole story. It's this beautiful collaboration between the machine and the human mind. The scientists, the experts, they step in to review everything, to make corrections, and to use their deep biological knowledge to make sure that the final book is as accurate as it can possibly be. They're the expert editors making sure the story is told right.
So that kind of leaves us with this incredible thought. With every new genome we sequence and annotate, we're adding another volume to the library of life. From weird microbes in the deep sea to exotic plants in the rainforest, we are just beginning to read the countless stories they hold. The only question is, what amazing new stories are we going to discover next?